Okay, there's nothing wrong with your screen. This is going to be a short video, hopefully short, where I tie together the adaptive immune response. It's the hardest thing is to see the big picture of this, and I realized too late that I didn't put in the slide that spells out the big picture for you. So that's going to be slide 47. That's what I'm going to show you now. So we can see the whole big picture of the adaptive immune system when we look at the situations where MHC systems are used. And in particular, the interactions between the cells. We can kind of anthropomorphize these. Think of whatever analogies you want. But there are a limited number of interactions that can happen that are necessary to trigger immune responses, and they're necessary to activate cells. And you'll see they're kind of different from each other. So the way these are spelled out is I show which MHC system it is, then I'll write which two cells are interacting with each other, and then the arrow, and then what happens if the T cell receptor recognizes a protein. So MHC is always displaying a protein to some kind of T cell. So what happens if the T cell does recognize that protein? If it doesn't recognize the protein, nothing happens. The T cell says, never mind, and moves on. But if it recognizes that protein, things will happen. And that's a big event. So these are the 5 situations. Okay, so the first one is with MHC1. A dendritic cell can show a protein it found to a naive CD8 positive T lymphocyte. So this would happen in a lymph node or in the spleen or any lymphoid organ. This is possible to happen. But typically we would think of this as a lymph node. So it's going to show a protein to a naive, so that is educated but not yet activated, CD8 positive T lymphocyte. That lymphocyte has been waiting its entire life, doing nothing. Well, if it recognizes this protein, it has now seen its purpose in life, and it will activate and become a CD8 positive cytotoxic T cell, which is an active cell that's going to run around the body and kill infected cells. So this is cytotoxic T cell activation. Those same activated cytotoxic T lymphocytes are going to look at MHC1 from other cells in the body. So that MHC1 is going to be used by other nucleated cells to show their cytoplasmic protein to the cytotoxic T cells. And if the cytotoxic T cells recognize that protein, they know that that nucleated cell is infected with whatever the thing is that's fighting the body, and that T cell will kill it. Kill that cell. That's what cytotoxic T cells do. The next one is activation of helper T cells, and that happens when a dendritic cell is going to show some protein it found to a naive CD4 positive T lymphocyte. This is going to happen in a lymph node, most likely. And that CD4 positive T cell has been waiting its entire life, doing nothing. But if it recognizes the protein shown to it on that MHC molecule, it knows its purpose in life, and it will become activated. It will make descendants that are CD4 positive helper T cells. And these helper T cells could be type 1 or type 2. So let's look at what those cells do. So those cells are going to use their T cell receptors to look at MHC2 in other contexts. So a macrophage, for example, is going to show what it found in its phagosome to a type 1 helper T cell using MHC2. the helper T cell looks at everything using its T cell receptor. It looks at that MHC2, and if it recognizes that MHC2, this helper T cell is going to send signals to the macrophage and say, yes, you did find the thing that is infecting us. Kill it, and the macrophage will become much more aggressive. This is really essential to a lot of situations where we have pathogens in our tissues. A lot of pathogens try to prevent this from happening. Listeria monocytogenes, for example, if this doesn't happen, we can't control it, and that's why it's dangerous to pregnant women, because they lose some parts of this response. And then B cells do this too. So B cells have B cell receptors, and they're going to occasionally detect something with their B cell receptor. And they are going to bring that in and digest it and put it on MHC2 and show it to a type 2 helper T cell. And if that helper T cell is seeing on that MHC2, if it's seeing the only thing it's able to see, the only thing its T cell receptor is good for, then it knows that B cell has found the thing that is infecting the body. That is, that helper T cell knows that that B cell is the right B cell to make antibodies against the thing that is infecting the body. So it will send signals to the B cell to send it through full-blown activation. And this is actually what I showed you on slide 39. So if we go back to slide 39, here we are. This is primary activation of a B cell, but it's also the full activation. This is what we call T cell dependent activation. I didn't put that here because that would be confusing, but a step that is not shown here is these presenting this to type 2 helper T cells. If that doesn't happen, they won't make memory T cells. So it's possible for B cells to do a kind of a weak activation without helper T cells, but they don't make memory cells and they don't make as much antibody. So it's unfortunate that the adaptive immune system is complicated enough that we can't always put everything in one picture, but understand that this would involve a helper T cell. Okay, so this is adaptive immunity, and if you can understand everything on this slide, which I obviously will take work because it's a lot of stuff to keep straight, but if you can put this on a table, draw a concept map or something, you will have the big picture of the adaptive immune system. There are endless details to put together, but these are the things that happen that determine whether we get an immune response or not. And if you know these things, you can figure out the probability of a strong immune response. So this is a concept map of adaptive immunity. And these are all the major events that might happen, and they start up here with some kind of antigen-presenting cell, like the dendritic cell I mentioned, stimulating T cells. And these would be CD4 positive, CD8 positive. These will become cytotoxic. These will become helpers. So cytotoxic T cell goes through its activation and makes memory cells. Helper T cell goes through activation, makes memory cells. The other thing they're going to do is they're going to actively run around doing their jobs, like being a cytotoxic T cell, killing virus-infected cells. These helper T cells, if they're type 1, they're going to stimulate macrophages. If they're type 2, they're going to stimulate B cell activation. So the B cell has had to find its antigen and present it to the type 2 helper T cell. And if that all works out, it is going to go all the way through making memory cells and plasma cells. That's all the stuff in blue. The stuff in red is memory cell secondary activation. So if somehow there's a second exposure Well, those memory cells can make all these different T cells, all these different plasma cells much faster. So that is, that's one way to draw out the entire adaptive immune system. And that's, to me, the most important thing you can take from these lectures is, what are all these cells doing? That helps you understand why why HIV is so bad, because HIV kills helper T cells. So if we're looking at CD4 positive T cells per cubic millimeter of blood, someone with HIV will, over the years, lose them until these can't happen. And, well, these can't happen, and that means B-cell responses can't happen, macrophages can't become more aggressive, and that leads to an early death. You can also understand why we don't have constant activation of our adaptive immune system. You have, I don't know the number, but at least 1 billion different T cells and B cells. What are the chances that one of those B cells is ever going to find the correct epitope? These are they have randomly generated receptors. So chances that any B cell is ever going to see its epitope are tiny. And until that happens, it will just wait. In order to have a B cell response, that B cell has to have seen that epitope and present it to a T cell that has already been activated using that same epitope because a dendritic cell found it somewhere else in the body. So these are insanely improbable events, and the only time they're going to happen is during a major infection. That's the big picture of adaptive immunity. The last thing I'm going to do in this video is skip ahead to, well, I have a slide that defines vaccines, and these are fundamentally a adaptive immune response thing. They trigger primary activation and memory cell formation so that when you're exposed to the actual pathogen, you get secondary activation. Some of the slides at the end of these at the end of this slide set show you how vaccines are complicated because sometimes they're going to make type 1 helper T cells when we need type 2 helper T cells. That sort of thing is hard for us to control. And so we don't know until we test it whether the vaccine is going to be useful or not. And I can tell you for things like tuberculosis, there are a lot of research labs all over the world generating candidate vaccines all the time. But getting the immune system to respond in the best way to that vaccine, like in the way that would prevent tuberculosis, is nearly impossible. So understanding how vaccines work is one thing. Understanding how to make a useful vaccine is a very different thing. There are a lot of other slides at the end of this about vaccines, a lot of slides about herd immunity and stuff. You're going to see all of those have the label on them, this label, because I'm not going to test you on them. So you can skip them if you want to, if they have this label. Okay, I'll see you at the next video.